You scoop water from a puddle, looks empty, clear, harmless. Put that drop under a microscope and a hidden city explodes into view. Thousands of creatures moving, hunting, regenerating, surviving in ways that make science fiction look boring. Some shrug off radiation that would melt you. Others cut themselves in half and walk away twice. A few spin like planets, coordinating thousands of tiny oars just to eat breakfast. Which ones are real? What can they actually do? And why does a single drop contain more biological tricks than most animals will ever learn? If you value documented microscopic biology and water ecology like this, subscribe. It's the best way to support our work. Also comment from which city you're hearing this from. This script is researched and factual. Using sources from eLife Journal published July 2024 on tardigrade DNA repair mechanisms, Current Biology from February 2017 on stentor genomics, and the Journal of Plankton Research's 2013 review by Jacek Radzikowski on aquatic invertebrate dormancy. These are peer-reviewed scientific publications, not Wikipedia summaries. Now back to the information. Number 1, tardigrade, water bear, moss piglet, call it what you want. This 8-legged microanimal walks through your backyard and survives conditions that would atomize you. 50 micrometers to 2 millimeters long. That's the size range. Plump body, 8 clawed legs. First described by German pastor Johann August Ephraim Goes in 1773. Italian biologist Lazzaro Spallanzani gave it the name tardigrada, slow stepper. Over 900 species confirmed. Fossils from the Cambrian period 500 million years ago. Tardigrades live in moss, lichen, leaf litter, soil, freshwater, and marine sediments, suck fluids from plant cells, animal cells, bacteria, get eaten by amoebas, nematodes, other tardigrades, standard food chain stuff, but when conditions turn bad, tardigrades do something no human, no mammal, no vertebrate can touch. They shrivel into a structure called a ton, lose 98% of their body water. Humans lose 1 to 4% and we're cognitively impaired. Tardigrades drain themselves to 3% water content and wait days, months, years, decades. In 1983, researchers collected moss from Antarctica, stored it in a freezer. 30 years later, they thawed it. 2 tardigrades woke up. One laid 19 eggs over 45 days. They called it Sleeping Beauty. 1, temperature tolerance, negative 272.8 Celsius. That's 1 Kelvin, nearly absolute zero. Paul Rom documented this in 1923. Upper limit, 150 Celsius for 15 minutes. Rom again, 1924. Pressure resistance. Tardigrades handle 600 megapascals. That's 6,000 times atmospheric pressure. 6 times deeper than the Mariana Trench. Ono and colleagues, 2008. Radiation. This is where it gets strange. Lethal dose for humans sits around 4 to 5 grays of ionizing radiation. Tardigrades, try 5,000 grays. A study published in Nature Communications, September 2016, identified a protein called DCUP, damage suppressor, coats tardigrade DNA, provides resistance. When researchers introduced DSUP into human cell lines, it suppressed DNA damage from X-rays by almost 40%. Thomas Boothby at University of Wyoming published in Communications, Biology, November 2022, explaining how tardigrades use a sugar called trehalose, working with proteins to survive desiccation. But the real mechanism goes deeper. Research published in eLife July 2024 by Marwan Anoud, Emmanuel Delagout, and Quentin Hello at Muséum National d'Histoire Naturelle in France revealed tardigrades don't prevent DNA damage. They repair it, massively. When exposed to radiation, tardigrades accumulate the same DNA breaks as human cells. But here's the trick. They upregulate DNA repair genes to extreme levels. These gene products become some of the most abundant proteins in the animal. The team discovered a tardigrade-specific gene they named TDR1. Tardigrade DNA damage response. 1. This protein doesn't gather at damage sites like normal repair proteins. Instead, it binds fragmented DNA and forms aggregates, compacts the shattered genome, maintains organization while other repair machinery stitches it back together. Space vacuum. European Space Agency's Photon M3 mission, 2007. 3,000 tardigrades launched to low Earth orbit, exposed to space vacuum for 10 days. Pressure 10 to the negative 6 pascals. Cosmic radiation 100 milligrays. UV radiation 7,900 to 7,500 kilojoules per square meter, depending on spectrum. Samples protected from light survived fine. UV-exposed tardigrades died in high numbers, but some survived UVA and UVB radiation combined with vacuum. First animal species ever documented doing that. Jensen and colleagues published results in Current Biology 2008. A November 2024 study in Nature by Chinese researchers, led by Lingqiang Zhang at Beijing Institute of Lifeomics, identified a new species hosting thousands of genes that activate under radiation. One gene, called DOTA-1, enables tardigrades to produce betylene pigments. Antioxidants. They mop up reactive chemicals that radiation creates inside cells. When researchers treated human cells with tardigrade betylens, survival improved dramatically. Most recent research, April 2024, from Bob Goldstein's lab at University of North Carolina, Chapel Hill, confirmed tardigrades ramp up DNA repair gene production to levels humans cannot achieve. Courtney Clark-Hachtel and Goldstein published in Current Biology, showing this response is the secret to extreme survival. The findings align with independent French experiments running simultaneously. University of Wyoming work suggests applications for pharmaceuticals and crop engineering. If we understand how tardigrades confer drying resistance, we might stabilize medicines without refrigeration, engineer crops for drought, protect astronauts from radiation on Mars missions. A microscopic blob with 8 legs that survived 500 million years might teach us how to survive the next 100. Number 2, Paramecium, the slipper, genus of single-celled ciliates, 50 to 300 micrometers depending on species. Covered in roughly 4,000 cilia arranged in longitudinal rows, each cilium beats 15 to 45 times per second. But cilia in the peristomal region? Double that, 30 to 90 beats per second. The cilia coordinate into metachronal waves, like wind across wheat. Propels the cell forward while it spins on its axis approximately 1 rotation per second. Antony van Leeuwenhoek likely observed Paramecium in late 1600s. Christian Huygens clearly described them in a letter from 1678. They became the white rats of phylum Ciliophora, found in freshwater, brackish water, marine environments worldwide, stagnant basins, ponds, anywhere organic matter decays, feed on bacteria, algae, yeast, sweep food through an oral groove using cilia. Food enters a buccal cavity, passes through cytostom, collected into food vacuoles that circulate through cytoplasm via streaming movement called cyclosis, speed. A study published in Scientific Reports, June 2021, by researchers measuring swimming speeds documented Paramecium aurelia, swimming at velocities strongly affected by viscosity, but not temperature, between 5 and 20 Celsius. The organisms are 50 to 320 micrometers, small enough to be seen with basic microscopes, large enough to track reliably. If a Paramecium scaled to human height, it would swim 4 times faster than Olympic gold medalists. That's proportional thrust. Accounting for viscosity at microscopic scales, behavior, when Paramecium hits an obstacle, ciliary beat reverses, swims backward briefly, turns, resumes forward progress. This is the avoiding reaction, triggered by calcium-based action potential. Some researchers call Paramecium a swimming neuron, single cell, no nervous system, but electrical signaling, calcium channels, sensory response, motor coordination, machinery that looks like neural function compressed into one cell, the genome of Paramecium tetraurelia sequenced, revealing 3 whole genome duplications. Some ciliates reassign stop codons, UAG and UAA code for glutamic acid and Paramecium instead of stop. Only UGA functions as stop. Temperature regulation exists. When cooled, calcium currents slow, action potentials broaden. Cilia reverse with longer latency. But over hours, Paramecium adapts. Behavior returns to normal. The new temperature becomes preferred. This is cellular learning without a brain. Research published in a PLOS Computational Biology. February 2023 modeled Paramecium as an electrophysiological and kinematic system. Swimming involves velocity, rotation angle, and spinning rate, all coupled to calcium concentration. The cell navigates chemically. Thermally? Mechanically. A study in results and problems in cell differentiation. March 2022 detailed how basal bodies anchor cilia, how ion channels control beating, how damage to anchoring proteins destroys coordination and motility collapses. Paramecium has a dual nuclear apparatus. Polyploid macronucleus, diploid micronucleus, reproduces asexually by binary fission, can conjugate sexually, exchanging genetic material. During fission, macronucleus splits by amitosis. Micronuclei undergo mitosis. Cell divides transversely. Each daughter gets copies. Well-fed Paramecium buds every 2 days. A 2021 study in Scientific Reports established Paramecium swimming speed as a measurable indicator of water pollutants. 5 different organic and inorganic contaminants at varying concentrations around drinking water. Thresholds all affected speed significantly. Erythromycin, tetrachloroethylene, trichloroethylene, zinc chloride, copper sulfate. Object tracking algorithms measured changes in minutes. The ubiquity of paramecium in fresh and salt water makes them natural biosensors. No reagents, no specialized tools, just a microscope and movement. In weightlessness, paramecium swimming velocity increased 7.5% initially. Texas 27 and 28. Our rocket experiments documented this. After 3 minutes in microgravity, speed returned to horizontal swimming baseline. Paramecium uses gravity for spatial orientation. Negative gravitaxis swims against sedimentation. When gravity disappears, the organism shows residual orientation for 80 seconds, then randomizes. Upon return to 1 GE, gravitaxis restored within 1 minute, even after 2-hour clinostat runs. A drop of pond water contains dozens of paramecium, each one processing thousands of environmental signals per second, adjusting ciliary beat, calculating trajectories, avoiding predators, finding food, all without neurons, all in a single cell 1/5 of a millimeter long. Number 3, Hydra, freshwater polyp, phylum Cnidaria, class Hydrozoa, body length when extended, 30 millimeters maximum. Usually 10 millimeters, tubular body, ring of tentacles around mouth at one end, sticky petal disc at the other, named by Carl Linnaeus in 1758 after the Greek mythological beast. When you cut off one head, 2 grew back. Hydra does the same thing. Cut it in half. Each half regenerates the missing part. The head grows a foot, the foot grows a head. Abraham Tremblay discovered this in 1740. He found green polyp-shaped organisms in pond water, cut them to determine if they were plants or animals. Reasoning: amputation kills animals but not plants. Days later, both pieces regenerated completely. He observed rapid contraction upon touch, tentacles that moved, buds that separated, not plant behavior. In 1741, he wrote to René Antoine Ferchaud de Réaumur, confirming these were animals. Tremblay performed numerous regeneration experiments, obtained 7-headed monsters, inspired Linnaeus and Pallas to apply the name Hydra. Tremblay published Mémoires pour servir à l'histoire d'un genre de polype d'eau douce in 1744. His work foreshadowed modern experimental developmental biology. Hydra regenerates from tissue pieces, even from dissociated single cells that reaggregate. Cut transversely or longitudinally, slice into many segments, middle pieces form both head and foot. Regeneration occurs without cell division, morphollaxis, tissue reorganization. This follows positional value gradients, head activation and inhibition gradients, foot activation and inhibition gradients. The gradients run opposite directions. Where both are low, buds form. Regeneration timeline. Wound closes within 1 hour. Epithelial cells reorganize. No gross changes until 30 to 36 hours post-amputation. Then small tentacle buds emerge from the regenerating tip. Tentacles mature over next 24 hours. Complete process takes 72 hours. Cells in the body column continuously divide and migrate. Cells at base of tentacles take 4 days to reach the tip and slough off. Cells in the body column take 2 to 8 days to reach budding zone depending on position. 1T signaling pathway controls this. One of the morphogens guiding embryonic development, found in Hydra, conserved through evolution. In Hydra, WNTT proteins trigger canonical beta-catenin-dependent or non-canonical pathways. These guide pattern formation, head formation, oral labral axis, tentacle and bud development, cell proliferation. In humans, disrupted WNT canonical pathway changes, MYC gene expression. Contributes to cancers. Hydra shares at least 6,071 genes with humans. Compare that to Drosophila with 5,696 shared genes. C. Elegans with 4,571. Hydra is a better genetic match for studying processes relevant to human biology than the standard model organisms. Asexual reproduction dominates. Budding. Section of body wall and digestive cavity develop into bud. Bud grows into miniature adult, breaks away when mature. Well-fed Hydra produces new bud every 2 days. Sexual reproduction occurs under harsh conditions. Before winter. Poor feeding. Ovaries or testes develop from interstitial cells. Swellings in body wall. Eggs or sperm released. Daniel Martinez published in Experimental Gerontology, 1998, claiming Hydra are biologically immortal. No aging. A sexually reproducing Hydra vulgaris, Hydra viridissima, and Hydra oligactis polyps, observed individually for years, showed no senescence. But age. Oligactis shows inducible aging. When transferred from 18 to 10 Celsius to induce gametogenesis, budding ceases, exhaustion, degeneration. Paul Bryan documented this in 1953. Hydra genome sequenced, reveals bursts of transposable element expansion, horizontal gene transfer, trans-splicing, gene simplification parallel to life cycle simplification, published Nature, March 2010 by Chapman and colleagues, Hydra magnipapillata, genome shaped by 3 periods of transposon activity. Most ancient expansion at 0.4 divergence level, most recent at 0.05. Some Hydra species live symbiotically with chlorella algae. Green Hydra. The algae photosynthesize, provide sugars, feed on Hydra's waste, benefit from protection. Hydra with symbionts survive longer without food, not obligate except in Hydra polymorphous. If Hydra dies, chlorella persists as free-living algae. Symbiosis dissolves in complete darkness. Most chlorella disappear, though some persist at posterior end. Hydra used in ecotoxicology, water quality monitoring, developmental biology, stem cell research, regenerative medicine, tumor pathobiology, a freshwater polyp 10 millimeters long with no brain cutting itself in half for reproduction while potentially living forever. That's the occupant of your local pond. Number 4, Volvox, colonial green algae, spherical colonies, 500 to 50,000 cells depending on species, sometimes called globe algae. First reported by Antony van Leeuwenhoek in 1700, found it distinctive, easy to identify under microscope. Occurs in freshwater habitats worldwide, ponds, ditches, puddles, cosmopolitan distribution. Volvox diverged from unicellular ancestors approximately 200 million years ago, Triassic period. Genera like Gonium with 16 chlamydomonas. Like cells represent intermediate evolutionary stages. DNA sequences from 45 volvocine species suggest transition from single cells to undifferentiated multicellular colonies took 35 million years. Transition to differentiated multicellular organisms with germ-soma division took less time. Each colony is a coenobium, hollow sphere, single layer of biflagellate cells embedded in gelatinous matrix. Cells arranged radially at periphery. Flagella protrude outward. Coordinated beating propels colony through water. Rolling motion. That's where the name comes from. Linnaeus, 1758, classified them as Volvox, meaning fierce roller, initially placed in order Zoophyta with cnidarians and tapeworms, later recognized as algae. Individual cells resemble chlamydomonas, each with 2 equal flagella, cup-shaped chloroplast, single pyrenoid, eye spot for light reception, 2 contractile vacuoles at flagella base. Cells range from ovoid to spherical to star-shaped, connected by cytoplasmic strands. These connections form during cell division, enable cell-to-cell communication. Polarity exists. Cells at interior have larger eye spots. Grouped together, facilitate phototaxis, movement toward light for photosynthesis. If light too strong, Volvox moves away. Protects chloroplasts from damage. Reproductive cells grouped at posterior. Somatic cells, vegetative. Reproductive cells are gonidia, lack flagella, never contribute to somatic functions, entirely specialized for reproduction. Colony size varies. Volvox aureus around 500 cells. Volvox globator up to 60,000. Diameter ranges 0.5 millimeter to 2 millimeters. Large colonies visible to naked eye. Appear as green specks in pond water. Reproduction asexual and sexual. Asexual dominates. Gonidia divide successively. Form hollow spheroidal structure called plaquea. Then undergoes inversion. Entire colony turns inside out. Process crucial because at end of cell division, flagella point inward, useless for motility. Inversion moves flagella to exterior surface. Complex developmental process requiring coordination among cells. Sometimes compared to gastrulation in animals, Volvox carteri studies show inversion driven by specific cell shape changes. Begins at anterior in V, carteri, at posterior in V, globator. Cell movements orchestrated by positional cues. Similar mechanisms exist in human embryonic development, though far more complex. Sexual reproduction occurs under stress. Unfavorable seasons. Ova and spermatozoa replace gonidia. Strictly ogamous, male and female gametes. Species can be monoecious or dioecious. Monoecious species protandrous. Male gametes mature first. Dioecious species separate colonies. Male antheridia produce spindle-shaped biflagellate antherozoids. Female oogonia produce eggs. Chemotaxis attracts antherozoids to eggs. Fertilization produces diploid zygote. Zygote forms thick wall, maybe smooth or spiny, accumulates hematochrome. Orange-red pigment, this is perennial body. Survives winter, germination involves meiosis, 4 nuclei, 3 degenerate, remaining divides mitotically. Forms new coenobium, Volvox contributes to oxygen production, serves as food for aquatic organisms, especially rotifers. V. aureus can form harmful algal blooms, warm waters, high nitrogen content, multiply rapidly, deplete oxygen, affect water quality. Studies on Volvox inform understanding of multicellularity evolution, division of labor, cellular differentiation, cooperation versus competition at cellular level. Life cycle is haplontic, haploid in all active phases, diploid only in zygote. This contrasts with diplontic organisms like humans. Volvox genome sequenced reveals conserved photosynthesis genes, light response pathways, 8 photoreceptor genes highly expressed in somatic cells, animal type rhodopsin VR1 expressed in reproductive cells. Gene regulation responds to light quality, cell type specific, different mechanisms control transcript accumulation during development and environmental response. A colony of 60,000 cells rolling through pond water, each cell photosynthesizing, communicating. Coordinating flagellar beats, producing oxygen, growing daughter colonies inside. When stressed, switching to sexual reproduction, microscopic but visible, simple but organized. Individual cells in unified whole simultaneously. Number 5, stentor, trumpet, animalcule. Genus of ciliated protists, 400 micrometers to 2 millimeters long. Trumpet-shaped when extended, can contract into ball 1/6 total size. Named by Abraham Tremblay in 1744, initially misidentified as Hydra type, first described to Royal Society as Polypus. Minute water animal. Christian Gottfried Ehrenberg detailed Stentor malari in 1831. Name references, Greek herald in Iliad, voice powerful as 50 men, alludes to trumpet shape, booming oral apparatus, body covered with over 100 longitudinal stripes. Pigmented cortical granules, blue in Stentor caeruleus, green in species with chlorella symbionts, brown in Stentor stepatus aggregates. Stripes alternate with rows of cilia, organized by parallel microtubule ribbons running entire cell length. Anterior end has membranellar band. Long fused cilia beat metacrinously, create feeding currents. Posterior has holdfast, attaches to substrate. Stentor exhibits complex behaviors, phototaxis, learning, habituation, contracts in response to mechanical stimulation, escape response from predators. After repeated stimulation, stops contracting. This is habituation, form of learning, no nervous system, single cell, yet exhibits decision-making that resembles neural computation. Movement patterns, 2 types, looping, somersaulting, bends over. Attaches mouth and tentacles to substrate, relocates foot, or bends and attaches foot to new location. Can move several inches per day, generally sedentary, attached to aquatic plants, rocks, submerged debris, sometimes free swimming, planktonic behavior, feeds on bacteria. Small ciliates, detritus, creates vortex with membranellar band, sweeps food into buccal cavity. Flow velocity reaches 100 micrometers per second, double vortex flow pattern, similar to Vorticella flow fields near no-slip boundaries. Prey organisms trapped, drawn in, digested. Regeneration abilities, extraordinary. Cut Stentor into pieces. Each piece regenerates into whole organism. Fragment less than 1/100th volume of adult grows back completely. Studied since Thomas Hunt Morgan's work 1901. Vance Tarter's Comprehensive Research 1961. Modern molecular studies reveal mechanisms. Regeneration after oral apparatus loss, induced by sucrose shock. Process takes 8 to 9 hours. Stage 1, teardrop shape. No membranellar band, lasts 3 to 6 hours. Stage 2, membranellar band appears, grows, 3 to 6 hours after treatment, lasts 3 to 4 hours. Stage 3, oral primordium appears at posterior end of band, 6 to 8 hours after treatment. Band and primordium migrate toward interior, 1 to 2 hours. Complete regeneration. Characteristic trumpet shape restored. Transcriptomic analysis published eLife September 2021 showed hundreds of genes differentially regulated throughout regeneration. Many genes not directly associated with oral apparatus indicates whole cell reorganization. Not just local structure repair. Nuclear apparatus distinctive. Highly polyploid macronucleus, varies in shape, moniliform in some species. Bead-like chain, 10 to 20 nodes in Stentor malari, variform tubular in others, condensed resembling enlarged single nucleus in others. Partly explains regenerative ability, multiple genome copies distributed through cell, Any fragment can regenerate because it likely contains complete genome information. Micronucleus, much smaller, contains information for macronucleus formation, essential for conjugation. Sexual reproduction rare. Stentor genome sequenced. Published Current Biology, February 2017. Unexpected findings. Uses standard genetic code. Unusual for ciliates. Many ciliate species reassign stop codons to amino acids. Stentor retains UAA, UAG, UGA as stop signals. Suggests Stentor represents ancient lineage. Split before code diversification in other ciliates. Symbiosis with chlorella common. Green Stentor. Endosymbiotic algae densely scattered in endoplasm. Provides sugars. Host provides protection. Nutrients from waste. Not obligate, except possibly Stentor polymorphous. Symbionts allow longer survival without food. Not fully autotrophic. Chlorella persist as free living if host dies. Association dissolves in complete darkness. Studies reveal complex ciliary coordination. Metachronal waves emerge during regeneration. Coordination spans millimeter scales, larger than most single cells. Hydrodynamics and structural rearrangement interplay. Ring topology, important for long-range metachronism. Physical coupling through fluid, not just direct cellular connections. Stentor found in freshwater bodies worldwide. Some species in marine environments, dystrophic ponds, brackish water, littoral zones, adaptable, cosmopolitan. A giant single cell visible to naked eye that learns, regenerates, and coordinates thousands of cilia like an orchestra without conductor. Number 6, rotifera, wheel animals, microscopic aquatic animals, 0.1 to 0.5 millimeters, smallest multicellular animals, complete organ systems, digestive, excretory, reproductive, nervous, packed in body smaller than many single cells, named for corona, rotating wheel-like structure covered with cilia at head, cilia beat, create impression of spinning wheels, draw food into mouth. First observed likely by Antony van Leeuwenhoek, 1701. Documented animalcules that survived desiccation, resurrected when water added, bedelloid rotifer, Philodina roseola, discovery of highly resistant dormant state in aquatic invertebrate. Jacek Radzikowski's 2013 review in Journal of Plankton Research states dormant forms of planktonic invertebrates are among most highly resistant stages in whole animal kingdom. Rotifers have jawed mouth, mastax, grinding plates, hardened structures, chew food, complete digestive tract, mouth to anus, nervous system with brain ganglion, eye spots, sensory organs, protonephridia, flame cells, excretion and osmoregulation, reproductive organs, ovaries, some produce resting eggs, 3 classes, Bedelloidea, Monogonata, Saisonidea, bedelloid rotifers, most common. Found in humid environments worldwide. Soil, moss, freshwater. Males never observed in bedelloid rotifers. Reproduce exclusively by parthenogenesis, cloning. All females. All genetically identical within strain unless mutation occurs. Movement varied. Free swimming, or inchworm-like creeping along surfaces. Anchor to debris. Live inside tubes or gelatinous holdfasts, some sessile, some colonial, adaptable to microhabitat conditions. Survival strategy, remarkable. Bedelloid rotifers undergo anhydrobiosis, ability to survive without water. Lose 98% body water, contract into compact shape called ton, permeable to gases and liquids but metabolically inert. Can remain dormant for years, even decades. Add water, rehydrate, resume activity within hours. Embryos, juveniles, adults all capable. Rotifers resist ionizing radiation, similar to tardigrades. Repair DNA double-strand breaks efficiently. Mechanism not fully understood, but involves robust DNA repair pathways. University of Wyoming, research and French studies both document this. Rotifers filter feed, eat dead material. Algae, bacteria, other microscopic organisms, important components of aquatic food webs, break down organic matter, transfer energy up food chain, eaten by fish, larvae. Aquaculture uses rotifer cultures to feed larvae. Brachionis species common, cultured commercially. Some species tolerate extreme environments. Antarctic soil, survive 6 years at -80 Celsius, published by Newsham and colleagues 2006. High salinity, low oxygen, acidic conditions, temperature fluctuations. Rotifers adapt, longevity varies. Lifespan typically days to weeks in favorable conditions, but dormant stages last indefinitely. Resting eggs survive decades. Viable after 100 years in some reports, creates seed banks and sediment. When pond refills, eggs hatch, population explodes. Rotifer genome sequenced for several species. Adenata vaga, Bidaloid rotifer, genome reveals evidence of extensive horizontal gene transfer. Foreign DNA integrated from bacteria, fungi, plants. More than 8% genome from non-metazoan sources, unusual for animals. Contributes to stress resistance. Nikon Small World in Motion Competition, 2012. Rotifer video won prize. Captured rotating wheels. Transparent body, internal organs visible. Cilia beating, mastax grinding. Digestive tract processing food. Movement coordinated. Efficient. Beautiful microscopic architecture. Turtle rotifer Testudinella patina. Has 2 eyes. Lorica. Protective shell, transparent. Resembles miniature turtle. Swims through water scanning for food. Shell protects from predators, but flexible enough to allow movement. Rotifers found wherever there's water. Temporary puddles, ephemeral ponds, permanent lakes, mossy rocks, roof gutters, leaf litter, soil interstices, Antarctic lakes, hot springs, versatile, ubiquitous, 2,000 described species, many more undiscovered. Complete animal in ½ millimeter, mouth, brain, gut, excretory system, sensory organs, muscles, reproductive system, survives desiccation, radiation, freezing, resumes life after decades dormant, filter feeds, reproduces, contributes to ecosystem function. And it's smaller than most Paramecium. Number 7, amoeba, shapeshifter, blob of protoplasm, single-celled organism, no fixed form, moves by extending pseudopods, false feet, cytoplasm flows, cell moves forward, surrounds food particles, phagocytosis, encloses bacteria, algae. Detritus in food vacuoles, digests internally, excretes waste, amoeba proteus. Most studied species, 200 to 600 micrometers. Large for single cell, visible under low magnification, found in freshwater, slow-moving streams, ponds, bottom sediments, feeds on diatoms. Smaller protozoans, detritus, no permanent structures except nucleus, cell membrane flexible, constantly changing shape, can flatten, extend projections, retract, flow around obstacles, squeeze through tiny spaces, form defensive cysts when conditions deteriorate, movement speed. 10 to 15 micrometers per minute, slow compared to ciliates, but effective for its hunting strategy. Settles on substrate, extends pseudopods, tests environment, moves toward chemical signals from food, away from harmful stimuli, reproduction by binary fission. Nucleus divides by mitosis. Cytoplasm pinches. 2 daughter cells, genetically identical unless mutation. Rapid when food plentiful. Population doubles every few days. Sexual reproduction rare. Occurs under stress. Genetic recombination. Increases variation. Amoeba used extensively in cell biology research. Easy to culture. Large size allows micromanipulation. Studies on cytoplasmic streaming, phagocytosis, cell movement, nuclear transplantation experiments, demonstrating nucleus controls cell behavior. Classic experiments by John Gurdon, 1960s. Laid groundwork for cloning research. Nobel Prize, 2012. Dictyostelium discoideum, social amoeba, slime mold. Individual cells when food available. Aggregate when starving. Form multicellular slug, crawls. Forms fruiting body. Spore-bearing structure. Cells differentiate. Some become stalk. Lift spores high. Some become spores. Survive until conditions improve. Remarkable example of cooperation at cellular level. Other amoeba species parasitic. Entamoeba histolytica causes dysentery. Naegleria fowleri, brain-eating amoeba, enters through nose, migrates to brain, fatal, but most amoeba harmless. Ecological roles: break down organic matter, regulate bacterial populations, serve as food for larger organisms. Amoeba genome sequenced, reveals complex cellular machinery. Cytoskeletal proteins, motor proteins, signaling pathways, more similar to multicellular organisms than bacteria. Eukaryotic cell complexity present in single cell. Freshwater amoebae contribute to nutrient cycling. Consume bacteria. Release nutrients in digestible forms. Support higher trophic levels. Play role in maintaining water clarity. Balance microbial communities. Amoeba under microscope. Transparent. Granular cytoplasm. Clear ectoplasm, outer layer. Granular endoplasm, inner region. Food vacuoles visible. Nucleus prominent. Contractile vacuole pulsates. Expels excess water. Maintains osmotic balance. Pseudopods extend, retract. Cell flows like liquid, but controlled, directed, purposeful, simple organism, no organs, no fixed shape, but successful. Survived hundreds of millions of years, found worldwide. From Arctic to tropics, ocean to freshwater, soil to sediments, adaptable, efficient, fundamental to aquatic ecosystems. That's 7 organisms, but 1 drop of pond water contains hundreds more. Euglenoids with photosynthetic chloroplasts and eye spots, diatoms with intricate silica shells, nematodes, flatworms, copepods, ostracods, bacteria uncountable, archaea, fungi, viruses, each with survival strategies, each adapted to microscopic scale. Latest research published April 2025 in Clearwater's online journal emphasizes diverse microorganism presence, indicates healthy pond ecosystems. Paramecium, vorticella, rotifers, daphnia, euglenoids, desmids as positive indicators. Variety matters more than single dominant species. Balance reflects stable conditions. Ostracods, seed shrimp, bivalve shell, legs protrude, 0.2 to 30 millimeters, but most around 1 millimeter. 8,000 species, crawl on sediment, eat bacteria, detritus, algae growing on plants, crustaceans, related to shrimp though microscopic, daphnia, water fleas, planktonic crustaceans, 0.2 millimeters to 6 millimeters, visible to naked eye at larger sizes, drift in water column, filter feeders, eat algae. Reproduce parthenogenetically most of time. Sexual reproduction when stressed, important food source for fish, natural water filters, consume massive amounts of algae, hydra feeds on daphnia, paramecium eats bacteria, rotifers consume smaller protozoans, stentor filters microorganisms, food web complexity and single drop, predator-prey relationships, competition for resources, symbiotic associations, all microscopic, all documented. September 2020, study at Blades Biological Puerto documented common pond organisms, tardigrades and moss, paramecium in organic rich water, rotifers everywhere water exists, daphnia in slow-moving water, planaria flatworms regenerating from fragments. Each organism fills niche, each contributes to system function. Pond ecosystems support thousands of species per cubic meter. Invisible diversity, genetic diversity within species, behavioral diversity, microhabitat specialization, seasonal variation, daily vertical migration, temperature preferences, oxygen requirements, pH tolerance, creates complex three-dimensional habitat structure in seemingly simple water. Recent study reveals tardigrade-specific protein DOTA1 produces betylene antioxidants. November 2024, Nature publication, Linjiang team. When applied to human cells, radiation survival improved. This microscopic organism teaching us cellular protection mechanisms applicable to medicine, space travel, radiation therapy. Paramecium research informs understanding of cellular computation, how single cell integrates thousands of signals, makes decisions, navigates environment, without brain, without neurons, just ion channels, calcium signaling, coordinated protein activity, applicable to understanding neural development, degenerative diseases, cellular communication. Hydra research impacts regenerative medicine, stem cell biology, tissue engineering, signaling pathways, pattern formation, immortality mechanisms, cancer research, development of organoids, tissue repair strategies, all from freshwater polyp 10 millimeters long. Volvox studies reveal multicellularity evolution, how cells coordinate, divide labor, differentiate, communicate, balance individual versus collective, game theory at cellular level, cooperation strategies applicable to understanding cancer, tumor suppression, cellular policing mechanisms. Stentor genomics reveals unusual mRNA splicing, trans-splicing events, gene structure simplification, genome dynamics, provides insights into genome evolution, gene regulation, non-standard mechanisms, expands understanding of eukaryotic gene expression, rotifer horizontal gene transfer, challenges assumptions about animal evolution. 8% genome from foreign sources, bacteria, fungi, plants, DNA integrated, functional, contributes to adaptation, raises questions about genetic boundaries, species concepts, evolution of complex traits. Scientists at University of California, Irvine use Stentor to study wound healing at cellular level, how cells sense damage, reorganize cytoskeleton, restore function, without immune system, without specialized repair cells, just single cell putting itself back together. Mechanisms conserved in animals, relevant to tissue repair. Researchers at MIT study Volvox inversion as model for gastrulation. Simpler than animal embryos, same physical principles, cell movements, tissue folding, mechanical forces, computational models, test hypotheses. Predictions confirmed by Volvox observations, inform understanding of development, Max Planck Institute studies rotifer desiccation tolerance, molecular mechanisms, trehalose production, late embryogenesis, abundant proteins, glass formation, protects cellular structures, applied research on crop drought resistance, seed storage, pharmaceutical stabilization, food preservation, Japanese research on tardigrade, P protein, introduced into human cells, protects from radiation, 40% reduction in DNA damage, published Nature Communications, 2016. Follow-up studies, test applications, radiation therapy, reducing side effects, protecting healthy cells, space radiation shielding for astronauts, genetic engineering for Mars colonization, a 2007 experiment, European Space Agency, tardigrades sent to space, 10 days low Earth orbit, exposed to vacuum, cosmic radiation, UV radiation, retrieved, rehydrated, some survived, reproduced, first animal to survive space exposure, open field of astrobiology, questions about panspermia, life transfer between planets, pond water sample from North Florida, Nikon microscopy, documentation, contains nematodes, protozoans, annelids, crustaceans, dipterans, colinterrates, gastrotrix, rotifers, tardigrades. Single drop, dozens of phyla, thousands of individual organisms. Biomass comparable to larger ecosystems when scaled for size. Chatogaster worms, freshwater annelids, predators, hunt smaller organisms, transparent bodies, segmented, sati for movement, related to earthworms but microscopic, active hunters in sediment. Urocentrum, rotund ciliate. Bifurcated by two bands of cilia, tufted tail, fused cilia on posterior, spins on tail, swims in irregular spiral, relative of Paramecium, found in same habitats, loxophyllum, ciliate predator, hunts rotifers, other ciliates, wart-like protuberances, groups of trichocysts. Discharge mechanism, immobilizes prey, freshwater habitats worldwide, metapus, anaerobic ciliate, lives in oxygen-depleted sediments, Antarctic seafloor specimens documented, survives without oxygen, uses alternative metabolic pathways, unique among ciliates, Spirostomum, giant ciliate, some species visible to naked eye, up to 4 millimeters, elongated, contractile, beats cilia in coordinated waves, found in stagnant water, organic-rich environments, Philodina, bedelloid rotifer class, 2 ovaries, moves by swimming or inching along surfaces, males never observed, exclusively parthenogenetic, common in littoral habitats, shoreline vegetation, Brachionis, rotifer with semicircular shield, corona-covered, scrapes organisms into mouth while browsing underwater plants, common in aquaculture, raised as food for fish larvae, Hypsibius exemplaris. Tardigrade species, model organism, used in radiation studies, genome sequenced, published. July 2024, eLife, TDR, 1 gene identified, DNA repair mechanisms characterized, reference organism for extremophile research. Microscope technology advances reveal more detail. Differential interference contrast, phase contrast, fluorescence, microscopy, electron microscopy, cryo-electron tomography. Each technique unveils structures invisible before. Tardigrade cellular architecture, stentor cytoskeleton, paramecium ion channels, rotifer organ systems, Volvox cell connections, understanding deepens, live imaging techniques, high-speed video, time-lapse microscopy, track cellular processes in real time, ciliary beating, pseudopod extension, cell division, regeneration, movement patterns, behavioral responses, data collection automated, machine learning analyzes movement, identifies species, measures health indicators, pond water, biodiversity assessments now use eDNA, environmental DNA, water samples analyzed, DNA sequences identified, species presence confirmed without visual identification, reveals rare organisms, cryptic species, difficult to see under microscope, expands knowledge of pond communities, climate change impacts pond ecosystems, temperature increases, altered precipitation patterns, drought, flooding, affects microscopic communities. Some species expand ranges, others decline. Monitoring programs track changes, rotifers as indicators, sensitive to pollution, temperature, oxygen levels, early warning systems, urban ponds, agricultural runoff, industrial contamination, all affect microorganism communities. Paramecium swimming speed decreases with pollutants. Rotifers disappear from toxic water. Daphnia populations crash. Hydra, tentacle damage from salt exposure. Biological indicators cheaper than chemical testing. Faster results. Integrated assessment. Conservation efforts focus on maintaining healthy pond ecosystems. Biodiversity. water quality, habitat structure, vegetation, substrate complexity, all support microorganism diversity, which supports larger organisms, fish, amphibians, birds, mammals. Foundation of food web invisible to most people. Education programs use pond water microscopy. Introduces students to biology, evolution, ecology, cell structure, physiology, behavior. Accessible. Requires minimal equipment. Pond nearby. Microscope. Slides. Students observe living organisms, ask questions, generate hypotheses, science in action, public engagement, citizen science, pond monitoring projects. Community members collect samples, identify organisms, submit data, contributes to research, builds scientific literacy, connects people to local ecosystems, appreciation for invisible biodiversity, art inspired by microscopic life, Ernst Haeckel's illustrations, 19th century, Kunstformen der Natur, art forms in nature, detailed drawings, rotifers, radiolarians, diadems. Influenced Art Nouveau movement. Contemporary artists continue tradition, microscopy, photography, video, digital art, beauty at microscopic scale, philosophy implications. What is organism? Volvox, colonial or multicellular, hydra, individual or superorganism with symbionts, amoeba. Where does cell end and environment begin? Boundaries blur at microscopic scale, definitions challenged, cognitive science questions. Stender learns without neurons. Paramecium makes decisions without brain. How? What is learning? What is cognition? Cellular information processing. Memory at molecular level. Challenges, anthropocentric definitions, origin of life studies, early Earth conditions, microscopic organisms, first life forms. Single cells persisted billions of years before multicellularity. Simple but successful. Pond ecosystems model primordial environments. Studying microorganisms informs understanding of life's origins. Astrobiology applications, tardigrade, survival in space, implications for life elsewhere, Mars, Europa, Enceladus, Titan. If microscopic Earth organisms survive space, could life transfer between worlds? Could extremophils colonize other bodies? Search strategies informed by terrestrial biology, synthetic biology, engineering organisms with tardigrade genes, drought-resistant crops, radiation-resistant bacteria for bioremediation, medical applications, tissue preservation, vaccine stabilization, inspired by nature's solutions, microscopic organisms as biotechnology sources, nanomedicine, drug delivery, biomimetic robots, inspired by ciliary coordination, paramecium propulsion, amoeba flexibility, stentor contractility. Engineering at microscopic scale learns from biological systems. Millions of years of evolution, tested solutions, a drop of pond water, millimeter scale cosmos, hundreds of species, thousands of interactions per second, energy flow, nutrient cycling, predation, reproduction, death, birth, all happening while you read this sentence. Invisible, unnoticed, essential. The late August 2023 study by Blades Biological emphasized simple truth. Pond life under microscope reveals complexity rivaling anything visible scale. Creatures, surviving conditions impossible for humans, regenerating from fragments, living potentially forever, coordinating thousands of parts in single cell, swimming 4 times faster than Olympic swimmers proportionally, making decisions without brains. We look at water, see nothing. Put it under glass, a hidden universe appears. Every pond, every puddle, every moss patch, entire ecosystems functioning, species interacting, evolution happening, survival strategies tested daily, microscopic arms, race between predators and prey, chemical defenses, escape responses, camouflage, speed, armor, toxins, all at scales measured in micrometers. Recent molecular studies showed genetic diversity within pond samples, rivals tropical rainforests when accounting for size, more species per unit volume, higher metabolic rates, faster generation times, rapid evolution, real-time adaptation observable. Laboratory cultures evolve new traits in months, demonstrate principles taking years in larger organisms. In 1973, Gozzi described tardigrades. In 1744, Tremblay published hydra work. In 1700, Leeuwenhoek reported volvox and rotifers. In 1678, Huygens described paramecium. Centuries of study, still discovering new species, new behaviors, new mechanisms. Microscopic world remains frontier. Technology improves. Understanding deepens. Questions multiply. Each answer generates more questions. How does single cell learn? How does organisms survive absolute zero? How does DNA repair itself after shattering? How do thousands of cilia coordinate without central control? How does colony behave as individual? The answers matter. Not just academically, practically, medically, technologically, philosophically. These organisms survived mass extinctions, climate changes, predators, parasites, competitors, continued for hundreds of millions of years. They know something about survival, about adaptation, about persistence, written in their genes, observable in their behavior, available for study in every pond. You walk past puddles, drive over ponds, ignore ditches, underneath surfaces, invisible to naked eye. Life thrives, complex, diverse, ancient, successful, waiting for microscope, waiting for curiosity, waiting to teach us what survival really means at most fundamental level. One drop, entire world, 7 creatures, infinite lessons,